Hi, this is Hristo and Emily, and you are listening to the MAPASGEN podcast.
We are so glad to have you with us today.
Yeah, welcome. Welcome to the room. We've got a really intense deep dive for you today.
We really do. It's one of those topics that it just kind of completely changes how you view things.
Exactly. I mean, imagine this. Imagine a single man walking right into a clinic.
Right.
He takes all the tests, passes every single medical check they throw at him, and donates his sperm.
And he doesn't just do this once, you know.
No, not at all. He does this continuously and ultimately helps bring a minimum of 197 children into the world.
Which is just a staggering number on its own.
It is, and he does this over the course of 17 years.
Yeah, almost 2 decades.
Right. And he crosses borders, into 14 different countries. That is 14 distinct European healthcare systems.
All with their own regulations and checks.
Exactly. And in every single clinic, he is handed a completely pristine, clean bill of health.
They tell me he's perfectly fine.
But, and here's the really terrifying part, hidden deep down within 20% of his reproductive cells is a ticking biological time bomb.
Yeah, a massive one.
A mutation that basically guarantees a near 100% risk of early onset cancer.
It's just chilling when you put it like that.
And it was completely invisible to every single standard medical protocol he encountered across all those years and borders.
Completely invisible. And that right there, that is the core mission of today's deep dive.
Yeah.
Because we really need to dissect this exact case. We're going to examine the systemic vulnerabilities that allowed a donor to just breeze through 14 national medical registries undetected.
Unbelievable.
Right. And we need to look at the specific biology of why standard blood tests fail, like why they totally miss localized genetic anomalies.
Because they clearly do.
They do. And finally, we're going to talk about how the current medical paradigm, which relies heavily on, reactive symptom-based treatment, is just fundamentally unequipped to handle the reality of human genetics.
Unequipped is putting it mildly, I mean.
Yeah, fair enough.
I just can't get over the timeline. 17 years.
It's a massive span of time.
That is an enormous span for a blind spot to just exist in modern medicine.
Yeah, it really is.
Like, we aren't talking about some minor clerical error, right? Like a lab tech losing a vial over a long weekend or something.
No, not at all.
17 years, that represents almost 2 generations of evolving clinical guidelines.
Oh, easily. I mean, you have entirely new generations of medical professionals entering the workforce during that time.
Right. And major updates in diagnostic technology.
Huge technological leaps, yeah.
And yet, despite all of that, this catastrophic genetic risk just remained completely undetected across the board.
Well, and when you map that 17-year timeline against the geographical spread...
14 countries.
Exactly, 14 different countries. It just exposes this massive structural flaw in how we define a universal medical standard.
Yeah, what does that even mean anymore?
Right.
Yeah.
Because we operate under this assumption that crossing a border into a highly developed European healthcare system, well, that it provides a redundant layer of safety.
Like a safety net.
Exactly. The logic is, if one clinic somehow misses something, the next country's regulatory framework will definitely catch it.
But, I mean, a redundant layer of safety is completely useless if every single layer is looking for the exact wrong thing.
Yes, exactly.
Like, if 14 different ministries of health are utilizing the exact same flawed diagnostic parameters, the number of borders crossed doesn't mean anything.
It's just repeating the same mistake.
Right. The risk doesn't go down, it just scales exponentially.
And that compounding nature of the risk, that's exactly what led to nearly 200 children being conceived.
Wow. 200 families.
Yeah. These are families who, they engage with the professional medical infrastructure. They assumed the protocols in place were totally comprehensive.
Because why wouldn't they?
Right. They trusted the system. They trusted that the biological material had been rigorously vetted.
They were entirely unaware that the system's definition of healthy was, frankly, dangerously narrow.
Dangerously narrow is a great way to put it. And to understand why it's so narrow, we really have to look at what this donor was actually carrying.
Okay, yes, let's get into that. The TP53 mutation.
That's the one.
This is the genetic error that causes what's called Li-Fraumeni syndrome.
Correct. Li-Fraumeni syndrome.
So let's really get into the mechanics of this for you listening. Because just throwing around the word cancer, it doesn't really capture the specific, brutal severity of this mutation.
No, it doesn't. To really grasp it, it requires looking at the actual function of the wild-type gene.
The normal one.
Right, the normal P53 gene. So in molecular biology, P53 operates as a transcription factor,
Okay.
and its primary role is genome maintenance. In fact, it is very often referred to as the guardian of the genome.
The guardian of the genome. That sounds pretty important.
It is incredibly important.
How?
Basically, when a cell undergoes stress, and we're talking specifically about DNA damage here,
Like from what?
things like radiation, exposure to toxins, or even just regular errors that happen during cell division.
Sure, just natural mistakes.
Yeah. So when that damage happens, the P53 protein is activated.
So it acts as like a highly specialized diagnostic tool within the cell itself.
Exactly that. It literally halts the cell cycle.
It just stops it.
Yep. It stops the cell from dividing, and it assesses the damage. Now, if the DNA can be repaired, P53 activates the necessary repair proteins to go fix it.
Okay, that makes sense.
But if the damage is too severe, if it's beyond repair, P53 triggers a process called apoptosis.
Apoptosis, that's programmed cell death, right?
Yes. It forces the irreparably damaged cell to literally self-destruct.
Wow.
Yeah, and it does that to prevent the cell from replicating its broken genetic code and spreading the error.
Okay, so if the P53 gene is mutated, if the guardian is broken, that entire diagnostic and repair pathway is just severed.
Completely severed.
The cell basically loses the ability to recognize its own structural failures.
That is exactly what happens. The mechanism of apoptosis is just disabled.
So the damaged cells just keep going.
Right. Without a functional P53 pathway, cells with shattered, highly mutated genomes don't die like they're supposed to.
Oh, man.
They just continue to divide, compounding their genetic errors incredibly rapidly.
And that's what forms a tumor.
Yes, that is what forms highly aggressive tumors.
Right.
So carrying a pathogenic variant in the TP53 gene, having Li-Fraumeni syndrome, it means this fundamental foundational defense mechanism is broken.
Which translates to what kind of risk for the person?
It translates to a near 100% lifetime risk of developing various malignancies.
Near 100%.
Yes, and it frequently strikes during childhood and adolescence too.
That is just heartbreaking.
We are talking about aggressive sarcomas, early onset breast cancers, leukemias, and brain tumors.
Just a horrific lineup of early onset oncology.
It really is.
And yet, with the near 100% risk of this, the donor was classified as healthy.
Over and over again.
This just brings us to a really hard look at the protocols themselves. I mean, if a man with a severed cellular defense mechanism passes all the standard checks.
We got perfect marks.
Right. We have to ask, what is the standard actually designed to protect against?
Well, it's a great question, because standard protocols for reproductive donors, they are deeply rooted in, 20th century medical paradigms.
So they're outdated.
Very. They were designed primarily to identify external transmissible pathogens.
Like catching a bug.
Yeah. The priority is screening for infectious diseases. HIV, hepatitis B and C, syphilis.
Okay, but what about the genetics?
Well, from a genetic standpoint, the standard protocol back then, and even often now, might just include a baseline karyotype.
Which is what, exactly?
It's essentially taking a microscopic picture of the chromosomes to ensure there are 46 of them.
Just counting them.
Pretty much.
Huh.
Just making sure there are no massive structural rearrangements or missing pieces.
Okay, so I want to use an analogy here because this is wild. It's like a macro-level structural audit.
Yeah, that's a good way to look at it.
It's like a mechanic giving a car a perfect safety rating because the paint job is flawless and it has 4 wheels, while completely failing to check if the brakes actually work.
That is exactly it.
They are confirming the building has the right number of walls, but they are completely ignoring the structural integrity of the concrete inside.
Right. They aren't looking at the microcode at all.
Just the macro.
Standard screening simply does not utilize high-resolution, next-generation sequencing to actually read the individual base pairs of the DNA.
So they just don't see it.
They don't. The protocol did exactly what it was engineered to do.
Which was catch the infections.
Right. It ruled out acute infectious threats. But it was fundamentally blind to a localized genetic anomaly.
Okay, but this brings up what I think is the most glaring mechanical question of this entire case.
What's that?
If Li-Fraumeni guarantees an incredibly high early onset cancer risk,
Which it does.
then how did this donor survive to adulthood without triggering some massive medical intervention?
It's a paradox, right?
Totally. How did he walk into clinics for 17 years, passing physicals and blood tests, without the mutation manifesting in his own body?
Well, the answer to that lies in the embryological timing of the mutation.
The timing.
Yeah. The reason the standard system missed it, and the reason his own blood work returned perfectly normal results, is due to a phenomenon called germinal mosaicism.
Yes. Germinal mosaicism. Let's unpack this for the listener because this is fascinating.
It really is the key to the whole mystery.
This is when the mutation isn't constitutional, right? Meaning it doesn't exist in every single cell of the body.
Exactly.
It's restricted to just a specific cellular lineage. Am I getting that right?
You nailed it. To really understand it, we need to trace this back to the donor's very first moments of embryonic development.
Okay, going way back.
Way back.
Yeah.
So the vast majority of genetic mutations are inherited, right?
Mm-hmm.
They are present in the zygote, which is the single cell formed right at conception.
From the parent.
Right. And because that original single cell divides to form the entire body, an inherited mutation gets copied into every subsequent cell.
So it's everywhere.
Exactly. But in this specific case, the donor did not inherit the TP53 mutation from his parents.
Really?
No. His original zygote was entirely healthy.
Oh, wow. So the mutation happened post-zygotically.
Precisely. Sometime during the very early cleavage stages of the embryo, as the cells were rapidly dividing.
Just days into development.
Yeah, very early. A spontaneous de novo mutation occurred.
Just a random error.
Yep. A random error in the TP53 gene of just one single cell out of the cluster.
And that specific timing is everything, isn't it?
It's the whole ball game. Because that one mutated cell, it continued to divide. And it created a lineage of cells that all carried that broken P53 pathway.
But the other healthy cells were dividing too.
Exactly. Meanwhile, the original healthy cells continued to divide, creating a completely distinct, healthy lineage.
So the donor developed into a biological mosaic.
Yes. A composite of two completely different genetic codes living in one body.
Man, biology is just crazy. And the critical failure point for the diagnostic tests, that's where those specific cell lineages ended up migrating during his fetal development, right?
That's exactly right. As the embryo differentiates and turns into specialized tissues, the lineage of cells that eventually formed his hematopoietic stem cells.
The ones that make blood.
Right, the cells in the bone marrow that produce his blood, those came entirely from the healthy lineage.
Wow.
So his blood literally contains zero trace of the TP53 mutation.
So when 14 different medical systems drew his blood for analysis,
14 different times, over and over.
they were just analyzing the healthy sector of his mosaicism.
Exactly. They were reading from a clean data set.
His blood was literally clean.
Totally clean. You could run his blood through the most sophisticated sequencing equipment on the entire planet, and it would return a negative result for Li-Fraumeni.
Because it just wasn't there to find.
Right. The mutation was localized elsewhere. It had segregated entirely into his primordial germ cells.
The reproductive system.
Yeah, the tissues that form the reproductive system. But even within his germline, the mosaicism persisted.
What do you mean?
Well, not all of his sperm carried it. Only an estimated 10 to 20% of his spermatozoa actually carried the mutated TP53 gene.
Only 10 to 20%.
Yeah. The remaining 80 to 90% carried the healthy, normal, wild-type sequence.
Okay, I have another analogy to clarify this because it's just so complex.
Go for it.
Imagine a massive library.
Okay.
The main catalog, which is like the blood test, says every single book in the building is totally safe to read.
Right.
But down on one specific shelf, completely hidden in the basement, which represents that 10 to 20% of the sperm, that shelf is just filled with corrupted pages.
That is exactly what it's like.
The system checks the catalog says we are good, so nobody even bothers to walk down to the basement to check the actual pages.
And taking that library analogy further, the danger isn't just that there's a shelf of corrupted books. It's what happens when one of those specific corrupted books is used as the absolute foundation to build an entirely new library.
Yes, the amplification of the risk.
Exactly.
Because for the donor, the mutation was just this isolated variable. He was basically navigating a localized risk.
Right, he was mostly healthy.
But when one of those specific mutated spermatozoa fertilizes an egg, the biological reality fundamentally shifts for the offspring, doesn't it?
It shifts entirely.
Yeah.
Because the fertilization process relies on that single spermatozoan delivering half of the foundational blueprint for the new embryo.
Right. The whole foundation.
So when a child is conceived from that specific 10 to 20% of mutated sperm, the TP53 mutation is no longer restricted to a little mosaic lineage.
It's not a localized basement shelf anymore.
No, it becomes constitutional. The child inherits the broken P53 pathway in every single cell of their entire body.
So a 10 to 20% localized risk for the donor just violently transforms into an absolute 100% whole body reality for the child.
A whole body reality.
The mutation is just baked in. It's in their bone marrow. It's in their organ tissue, their neurological architecture.
Everywhere.
From the absolute moment of conception, the child's cellular defense against cancer is just fundamentally deactivated.
It's deactivated from day one, which, forces us to really look at the tragic statistical fallout of this case.
Yeah, let's look at the numbers.
We're looking at a minimum of 197 children conceived from this donor.
197.
And if you apply that 10 to 20% mosaic mutation rate, statistically we're looking at dozens of children who possess a constitutional TP53 mutation.
Thousands of children.
Yeah.
We need to just slow down here for a second.
Yeah. Okay.
Because we have to shift from this abstract biological puzzle to the actual clinical outcomes on the ground.
The real families.
Right. We are looking at families who went through completely exhaustive fertility processes. They were relying heavily on medical guidance, only to discover this catastrophic flaw years after the fact.
And the tragic part is, they only discover the flaw when the biology forces its way to the surface.
When it's too late.
Exactly. The current data from this specific case indicates that 10 children have already been diagnosed with severe oncology.
10
pediatric patients currently undergoing intensive cancer treatments.
Just all due to a blind spot that existed right out in the open across 14 national borders.
Right. And considering the nature of Li-Fraumeni syndrome, the tumors these children develop are uniquely devastating.
Why is that?
Well, we established earlier that P53 triggers cell death when DNA is damaged. Right?
Okay, apoptosis.
But think about how traditional oncology treatments actually work. Standard chemotherapy and radiation therapy, they operate by intentionally inflicting massive DNA damage on the tumor cells.
Oh, wait, I see where this is going.
Right. They inflict damage, heavily relying on the cell's own P53 pathway to recognize that radiation damage and trigger apoptosis.
So the treatment relies on the very guardian that these kids are missing.
Exactly. The mechanical reason P53 mutated tumors are so incredibly aggressive is precisely because they resist standard interventions.
That is just awful.
When you apply radiation to a Li-Fraumeni tumor, you are damaging the DNA, yes. But without a functional P53 sensor to initiate cell death, the tumor cell simply absorbs the radiation.
It just absorbs it.
Yeah, it absorbs it, mutates even further from the radiation, and continues to rapidly divide.
So the standard treatment can actually make it worse.
It can. It completely forces oncologists to utilize highly specialized, totally experimental, and relentlessly aggressive targeted therapies instead.
And this clinical reality, this exact nightmare scenario, perfectly defines the failure of what we call the reactive medicine model.
It really is the ultimate example of it.
Because the entire structure of modern healthcare in so many ways is built around responding to a symptomatic manifestation.
Right, waiting for a symptom.
We wait for the tumor to grow large enough to cause pain or to appear on an imaging scan, and only then do we mobilize the medical infrastructure.
We wait until the house is already fully engulfed in flames.
Yes. We are mobilizing to fight a fully realized cellular fire rather than taking steps to identify the spark before it catches.
And the dual costs of this reactive framework, they're just completely unsustainable.
Let's talk about those costs.
Let's look at the direct financial impact first, just from a systemic level.
Okay.
Treating therapy-resistant pediatric oncology requires immense resources. We're looking at costs easily exceeding 100,000 euros per year per patient.
Over 100,000 euros a year.
Yes. And that covers intensive care unit stays, highly specialized surgical interventions, continuous advanced imaging, and these targeted molecular therapies that are often not widely subsidized by national health plans.
That is a staggering financial burden.
It is.
But, is the financial cost even the real tragedy here?
I mean, no, of course not.
Right. 100,000 euros is a massive number, but the true price is being paid in human lives. It's paid in just unimaginable family trauma.
Completely.
How can we, as a society, accept a medical model that literally waits for a child to get sick before looking at the underlying biological code?
We can't, or at least we shouldn't anymore.
It feels like a systemic abdication of responsibility, just waiting for a child's genome to fail before intervening.
And that profound systemic failure is exactly what's driving an urgent paradigm shift within the medical community right now.
It has to.
The reliance on this reactive symptom-based intervention is facing really heavy scrutiny, precisely because the diagnostic technology to prevent these outcomes It already exists.
We have the tools.
We absolutely do. Which brings us to the methodology utilized by advanced genetic platforms.
Like the MAPASGEN paradigm.
Exactly. The MAPASGEN approach represents a total, absolute rejection of the reactive model.
We are talking about a shift from assumption to actual data-driven prevention.
Right.
The core methodology here is what's called expanded carrier screening.
Yes. Expanded carrier screening.
But, okay, we have to address the obvious logic gap that you listening are undoubtedly thinking about right now.
The elephant in the room.
Right, because if standard blood tests missed the donor's germinal mosaicism because the mutation literally wasn't in his blood,
Right. We established his blood was clean.
then how does expanded carrier screening actually solve the problem? If a new advanced platform just draws his blood again, wouldn't it miss the exact same blind spot?
It's a great question, and it highlights the critical distinction between targeted legacy testing and next-generation sequencing, especially when combined with comprehensive tissue analysis.
Okay, break that down for us.
So when utilizing an expanded preventive framework, particularly for reproductive donors, the protocol shifts in two very fundamental ways.
What's the first way?
First, the screening relies on ultra-deep sequencing. Standard genetic tests often just look for a handful of common mutations using basic genotyping.
Just looking at the usual suspects?
Right. But expanded next-generation sequencing, or NGS, it reads the entire coding region of thousands of genes.
So, going back to the library analogy,
Yeah.
it isn't just checking the catalog to see if a specific book is on the shelf.
I know.
It is opening every single book and reading every single letter of the text to find a typo.
That is exactly it. It's reading the actual letters of the DNA. And the second fundamental shift, and this is how you specifically combat the threat of germinal mosaicism in a clinical setting.
Yes. How do you catch the mosaicism?
Advanced screening requires direct analysis of the gametes themselves.
The actual reproductive material.
Exactly. You simply cannot rely on a somatic blood draw to verify the integrity of the germline.
Because they might be completely different data sets.
Exactly. The genetic architecture of the spermatozoa must be sequenced directly.
So they test the sperm itself.
Yes. By analyzing the actual reproductive material, these platforms can identify the specific mosaic allele frequencies.
They can see the mutation in the 10 to 20%.
Exactly. They catch that 10 to 20% corrupted data before it is ever utilized for conception.
See, that distinction is just paramount. It is the difference between blindly hoping the system is safe and actually verifying the specific biological inputs.
Verifying the inputs changes everything.
But, we also need to be incredibly rigorous about what this paradigm shift actually promises.
We do. We have to be honest.
Right, because if a patient utilizes expanded carrier screening, they really need to understand the limitations of human biology.
Absolutely.
We are breaking down an illusion of safety today, so we really cannot afford to just construct a new, shinier illusion in its place.
No, and absolute transparency is the foundation of the preventive model. Like MAPASGEN insists on honesty over empty promises.
Because biology is chaotic.
It is inherently volatile. Expanded screening drastically reduces risk. It identifies known pathogenic variants. It deeply analyzes recessive carrier statuses, and it catches severe dominant mutations like TP53.
It maps out the known dangers.
Right. But no ethical platform is going to promise a flawless outcome.
You can't guarantee perfection.
As we discussed with the donor's original de novo mutation, spontaneous genetic errors can occur at the exact moment of fertilization.
Entirely independent of the parental DNA.
Exactly. You can have perfect gametes, and a mutation can still occur postzygotically.
The universe just maintains a baseline level of entropy. You just cannot control every variable.
You can't. But what you can do is eliminate the known, inherited, catastrophic variables.
You eliminate the preventable tragedies.
Yes. The preventive paradigm is all about providing comprehensive, actionable data. It illuminates the blind spots that legacy protocols just ignore.
Which gives families the power.
Exactly. It allows families to make decisions based on high-resolution biological reality, rather than just relying on statistical assumptions and hoping for the best.
It totally redefines the concept of freedom in reproductive medicine.
It really does.
Because, when people hear genetic screening, the immediate association is often this fear of over-engineering, right?
Like designer babies.
Right, attempting to design an impossible, perfect biological standard. But that fundamentally misunderstands the objective of this technology.
It completely misses the point. The objective isn't perfection. The objective is agency.
Agency. I like that.
It is providing the individual with informed choice over their own genetic legacy.
Think of navigating the fertility process under the legacy reactive model.
Okay.
It's like being asked to pilot a ship through a highly complex, reef-laden channel without a sonar system.
You're just sailing blind.
Right. You are moving forward, trusting that the water looks clear from the surface. You're completely blind to the jagged topography right beneath you.
And eventually, you're going to hit something.
Exactly. And when you inevitably strike a reef, the medical system sends out this highly advanced, incredibly expensive rescue fleet to try and save you.
And while that rescue fleet is obviously necessary at that point...
It doesn't change the fact that the hull has already been breached. The damage is done.
Exactly. True freedom isn't piloting the ship blind and just hoping you don't sink.
No. True freedom isn't walking into a dark forest and hoping there are no predators.
Right.
True freedom is possessing a high-resolution sonar map of the entire channel before you even leave the port.
Before you even pack your bags.
Exactly. Expanding the diagnostic criteria provides that map. It allows prospective parents to identify structural threats in advance.
Because once you have the map, you have choices.
Right. If a donor carries a high-risk pathogenic variant, you just chart a different course.
You choose a different donor.
Or if both partners carry a severe recessive mutation, you utilize targeted interventions like IVF with pre-implantation genetic testing.
Exactly. To ensure the resulting embryo does not inherit the disease.
You transition from being a passive recipient of biological chance to an active, informed protector of your family's future.
But embracing that role, it requires a significant cultural shift in how we view our own genetic data.
What do you mean?
Well, we have to move past the fear of uncovering our own biological anomalies.
A lot of people don't want to know.
They don't. But every single human being carries mutations. All of us.
Nobody has perfect DNA.
Nobody. Acknowledging that reality and actively utilizing advanced sequencing to map out those anomalies, it is literally the only way to build a proactive medical framework.
As we synthesize the layers of this deep dive today, the trajectory of this specific donor case serves as a really profound warning.
A wake-up call, really.
We analyzed a massive systemic failure, a 17-year period where standard protocols across 14 distinct countries operated with a glaring diagnostic blind spot.
And we examine the embryological mechanics of why that happened, germinal mosaicism.
Right, how a post-zygotic mutation can completely hide from somatic blood analysis.
Only to manifest with absolutely devastating certainty in the next generation.
We also detailed the clinical consequences of Li-Fraumeni syndrome, outlining exactly why relying on reactive medicine to treat P53-deficient tumors is just an agonizing strategy.
It's economically exhausting and ultimately flawed.
Right. And the transition toward a preventive paradigm, characterized by platforms utilizing comprehensive sequencing and direct gamete analysis, it is not merely an option anymore.
No, it is a clinical necessity.
It's the absolute baseline of modern reproductive care. And this is where the implications extend directly to you, the listener.
It is so vital to actively engage with your own genetic health.
Don't just take their word for it.
Exactly. Do not accept a standard clearance without understanding the precise parameters of what is actually being tested.
Demand comprehensive data.
Explore the methodologies of preventive platforms, question the legacy protocols, and utilize the advanced diagnostic tools that are available today.
Because genetic awareness is the base.
Genetic awareness is the base upon which all informed decisions are made.
Genetic awareness is the base. And before we close out this deep dive, I want to leave you with a final thought to examine.
Something to mull over.
Yeah. We have spent this time analyzing a single donor, right? A specific mutation in the P53 pathway and the nearly 200 lives fundamentally altered by a medical blind spot.
One case out of so many.
Exactly. Consider the sheer volume of biological material moving through clinical systems every single day.
It's enormous.
If a mutation carrying a near 100% cancer risk can quietly cross 14 national borders under the guise of standard healthcare...
What else is slipping through?
Right. What other silent genetic inheritances are currently being passed down simply because we haven't yet dared to demand to look deeper into our own biological curve?
It is a reality that requires immediate systemic reevaluation.
It absolutely does. Thank you so much for joining us on this exploration today.
We appreciate you listening.
We will catch you in the next deep dive.